Hello everyone. Welcome to lecture 2 on plant growth and development. So in the last lecture what we studied about the plant growth and its characteristics, right? So we studied that the plant growth is indeterminate. That is, there are certain cells like meristem cells that have the capacity to divide unlimitedly. And also we studied about two different types of growth in plant cells, which are arithmetic and geometric growth. So in arithmetic growth, we saw that it is a straight line, it follows a straight line equation where only one progeny cell further divides and the other progeny cell becomes differentiated and acquire a function. So only one progeny cell further divide, and in geometric growth, both the progeny cells divide. So in geometric growth, both the progeny cells divide, giving it a S-shaped curve. So a S-shaped curve is obtained in which the initial phase is lag phase, the middle phase is the exponential or the growth phase, and the third phase is the stationary phase. So the equation which is followed in arithmetic growth is L naught is equal to, LT, which is the growth at time t, which is shown on y-axis, and time is being shown on x. So LT, that is the growth at time t, is equal to L naught plus RT. And for the geometric growth, the equation is WT is equal to W naught ERT, e to the power R and T. Here R is also known as the efficiency index. So these were the two different types of growth that happens within the plant, and in different during the embryonic development, we can see both type of growth where the plant cells undergo geometric growth as well as the arithmetic growth. So in the early stages, plant cells follow the geometric growth. So initially the zygote, which is formed by the fusion of the male and female gametes, undergo rapid exponential cell division, and since it is a geometric growth, therefore each cell will divide further. Each progeny cell will divide further. So this is the zygote divided. Then each of these two cells undergo division. Similarly, the geometric phase is being followed. So 2 cells, then 4 cells. It is a 2D structure, but otherwise these are 8 cells, 16 cells, and so on. So geometric phases, all the cells divide, and each cell division doubles the number of cells, leading to the geometric growth. So in this, 2 into 2 is 4, 4 into 2 is 8. So at each division, the number of cells are getting doubled. And this phase is crucial for quickly building up the cell population. So since initially at the starting of the development, we start the development with only one cell, which is the zygote. So we need to create more cells rapidly for the development, and therefore this geometric growth is required at that stage. Later on, arithmetic growth pattern is being followed, and as the embryo develops and cells start to specialize and differentiate. So once the embryo is being developed, certain cells lose their capacity to divide, and therefore only certain cells are capable of division, leading to the arithmetic phase. And the growth pattern may shift to the arithmetic growth. From the geometric growth, it shifts to the arithmetic growth. Here, the addition of new cells or the expansion of tissue happens at a more constant rate. So in this case, the addition of new cells or the expansion of tissue happens both at a constant rate. because in this, these new cells, out of the two progeny cells, one will divide and one will become specialized and lose the capacity to divide. So both of these happen at a more constant rate. Now, how these dividing cells get specialized? So this process is known as the differentiation of the cells. So this process where the cells lose their dividing capacity and they acquire a particular function is known as the differentiation of the cells. What is cell differentiation? Differentiation is the process by which unspecialized cells, the cells which initially do not have any function, which are unspecialized to perform any function, become specialized into one of the many cell types that make up the plant. This process involves changes in the cell structure, function, and gene expression. So we know there are various types of cells that are present within the plant. So each of these cells have different cell structure. Each of these cells must be performing different function, and they must be having different proteins as a result of different gene expression. So the process where the unspecialized cells undergo change to form the specialized cells with particular structure, no particular structure, function. But the specialized cells have a particular change in the cell structure, they have a particular function and gene expression pattern. So the process of differentiation involves cell specialization and gene expression. So during differentiation, cells develop specific structures and functions that allow them to perform specialized roles within the plant. So this specialization is being acquired by the cells, and certain genes are being turned on or off, producing the proteins that determine the cell's structure and function. So therefore, the changes in the gene expression takes place so that the cells can perform a particular function. Examples where the differentiation of cells happen is xylem and phloem formation. So these examples are important that as a result of differentiation, xylem and phloem formation happens. So in the vascular system, some cells differentiate into xylem cells, which are involved in water transport. So xylem is involved with water transport, phloem is involved with the nutrients and sugar transport. So in case if you forgot, just remember water and xylem. W is close to X. When we study A to Z in alphabetical order, W is close to X. Similarly, if I talk about nutrient and phloem, certain people get confused between these two things. So nutrient and phloem. So nutrient N is close to P in alphabetical order. So water is close to xylem. So xylem cells are involved in the water transportation. So this is just a mnemonics for you to remember. And nutrient cells are and phloem cells are specialized to carry the nutrient transportation. So in the vascular system, some cells differentiate into the xylem cells, which are involved in water transport and structural support, while other cells become phloem cells responsible for transporting nutrients. So these changes happen as a process of differentiation. Next example is the formation of root hair cells. So in root, some epidermal cells differentiate into root hair cells, which increases the surface area for water absorption and nutrient absorption. So these are certain examples where the differentiation of cells happen and cells acquire a particular function within the plants. Then the next process is de-differentiation. De-differentiation. So as the name suggests, if I break it into two parts, de-differentiation means reversal of differentiation. That is, the cells are de-differentiated. That is, they lose in the differentiation, they acquire a particular function. So in de-differentiation, they will lose that function. and regain their capacity to divide. So de-differentiation is a process by which specialized differentiated cells revert to a less specialized. So from the specialized, they are reverting to the less specialized, more primitive state. And now they regain their ability to divide and produce different cell types. So this is de-differentiation, that is a reversal of differentiation. So the process in the differentiation, the process was they acquire specialized structures. So in differentiation, the process will involve the loss of specialization. So differentiated cells lose their specialized structures and functions, become more like their precursor or stem cells, where they undergo division. So for cell division, the cell has to enter into the cell cycle. Therefore, they re-enter into the cell cycle, gaining the capacity to divide and proliferate. So this is de-differentiation. In certain stages of the plant life, de-differentiation happens. Example, callus formation. So when a plant is wounded, you might have seen that the callus is being formed by the plant. So cells at the injury site de-differentiate to form a callus. These callus cells are undifferentiated and can proliferate to heal the wound or generate new tissues. Whenever the formation of new tissue is required, in that cases, differentiated cells have to undergo de-differentiation so that they can carry out cell division and can undergo proliferation to form the new cells or new tissues. The second example is the cambial cells. So in vascular cambium, cells can de-differentiate to form new meristematic cells. As a result of de-differentiation, new meristematic cells are formed which contribute to the secondary growth. So secondary xylem and phloem, formation of secondary xylem and phloem happens as a result of de-differentiation. Secondary growth is happening as a result of de-differentiation. Right? The third one is the re-differentiation. So as the name suggests, that is again differentiated. Re-differentiation. Again undergo differentiation. So redifferentiation is a process by which de-differentiated cells that have initially lose their structure and acquire the ability to divide, they again become specialized, but this time into a different cell type. This process is crucial for plant regeneration and development. So basically what I mean to say is, initially when the cell undergo differentiation, it acquire a particular structure and function. Suppose it is forming a cell type X. Now it undergoes de-differentiation. De-differentiation, where it does not have any specialized function, Now these de-differentiated cells again undergo redifferentiation. So these redifferentiated cells again acquire a particular function and will form a particular cell type. But this cell type that will be formed is different from the one which was earlier formed by the similar cells. So earlier, suppose it is forming the X cell type, upon redifferentiation, the cell type may change. So these three things are very important in development, where there is a coordination between the differentiation, de-differentiation and redifferentiation depending upon the conditions. So whenever there is a need to increase the number of cells, so differentiated cells undergo de-differentiation so that they can again enter into the cell cycle and can increase the number of cells, and can then further undergo redifferentiation, where they can generate tissues that are needed for the development. So specialization after de-differentiation is a process that happens So after dedifferentiation, cells undergo another round of differentiation to acquire new specialized functions. And this process is often involved in the formation of new tissues or organs from previously dedifferentiated cells. Examples is the regeneration of the shoots and the roots. So in tissue culture, this is important. This might be asked that in tissue culture, callus cells can redifferentiate to form new shoots and roots. So we carry out tissue culture using the callus cells, right? So callus cells are used in the tissue culture, TC, tissue culture, where they undergo redifferentiation, and these cells undergo redifferentiation to form new shoots and root, and this ability is utilized in plant propagation and genetic engineering. So these are the certain uses also of the mechanisms that happens within the cell. Formation of adventitious roots. So when a cutting is placed in the soil or water, the cut stem cells dedifferentiate. First, it undergo dedifferentiation and then redifferentiate to form new roots, enabling the cutting to develop into a new plant. So these are the three processes that happens at the cellular level: differentiation, dedifferentiation, and redifferentiation. Now, list of tissues in a. So there was a question in NCRT that enlists certain tissues that are the products of redifferentiation. So this is the list of tissues in a woody dicotyledonous plants that are the products of redifferentiation. So secondary xylem is a process of redifferentiation, secondary phloem, cork or phloem, and secondary cortex or phallodum. These are the products of the redifferentiation. Now we will study about the development in plants. So plant development, as I mentioned, is a highly coordinated process involving growth, differentiation, and morphogenesis, that is changes in the structure, regulated by genetic, hormonal, and environmental factors. So plant development is regulated at the genetic level as well, hormonal level as well, and the environmental factors as well. So hormonal, we will see plant growth regulators, PGRs, we will see later. This intricate process allows plants to grow, reproduce, and adapt to their environment, ensuring their survival and propagation. So the development involves these basic steps, where firstly the meristematic cells, which have the capacity to divide, shows plasmatic growth. That is, there is an increase in the amount of protoplasm. There is an increase in the amount of protoplasm. Not plasma, protoplasm. So it is a plasmatic growth. With the increase in the amount of protoplasm, the cells can undergo division, and the expansion happens, the maturation may happens, resulting in the formation of mature cells. These mature cells are being formed as a result of differentiation, expansion, maturation, and the mature cells undergo senescence. That results into the death. Senescence means old age, aging. So this is the sequence of the development process in a plant cell, that with the plasmatic growth, the expansion or elongation start happening, and the cells undergo division. That also results in differentiation, and ultimately the differentiated cells mature, resulting in the formation of mature cells. So these certain stages are overlapping, and this is how the development in the plants happen. So we will study these steps one by one in details. The first is the meristematic cell. They are found, we know that they are found in the meristem regions of the active cell division, like root and shoot apex, and in the apical buds. So they are found in the meristematic regions of the active cell division. Meristematic cells are undifferentiated cells. They are capable of continuous division. They give rise to various specialized cells that contribute to the growth and formation of different plant tissues. The second step involves the cell division. So meristematic cells divide by mitosis to produce new cells. These new cells can either remain meristematic, or they can begin the process of differentiation, as being shown in this figure. So they can either remain meristematic, like you can see here with the this has been shown that these that undergoes plasmatic growth, undergoes cell division, resulting in the formation of some meristematic cells, and certain will differentiate. So some cells will undergo again they will form the meristematic cells, and some will acquire a particular function. So outcome is they generate new cells for plant growth, repair, and development. The third steps involve the plasmatic growth. So this phase involves increasing cell size due to the synthesis of new cellular material and the cell cytoplasm enlargement. And it prepares the cell for subsequent stages of growth and development. So this is the plasmatic growth that promotes the cell for the subsequent growth and development. Fourth is expansion. So cells elongate by taking up the water and enlarging their vacuoles. Initially, we know that the vacuole is small and the nucleus is larger, but with time, the vacuolation increases. The cell wall is loosened to allow for this expansion. And what is the significance of this step? It is essential for the growth of plant organs such as stems and roots, enabling them to increase in the length. The next is the differentiation. So the process by which cell is specialized in structure and function. Example, differentiation leads to the formation of various types of cells, as I mentioned, xylem, phloem, epidermal cells, etc. Maturation, differentiated cells undergo final modifications to become fully functional. So this process is known as maturation. This includes the development of secondary cell walls in some cell types such as xylem cells. And outcome is that the mature cells perform a specific function essential for the plant's survival and growth. So these are the mature cells which are formed as a result of differentiation. And then what is a mature cell? So fully differentiated and functional cells that carry out specialized tasks. For example, mature xylem cells transport water and minerals from roots to the other parts of the plant. These cells perform their roles effectively until they undergo senescence, that is, until they become aged. So, senescence is the process of aging in cells leading to decline in function and eventually death. Signs that the cells will lose their functionality, they break down their cellular components and contribute to recycling of those nutrients within the plant. Senescent cells eventually die and the plants may reuse their components. So this is the aging, and ultimately the cells die, so which is the death of the plant cell. Final stage where the cell undergo programmed cell death, which is known as apoptosis, or die due to damage or aging, which is the necrosis also. Importance, this process is crucial for the plant development, such as in the shedding of leaves, flowers or fruits, and in the formation of certain structures like xylem vessels, which must die to function properly. So xylem has to carry the water. So these xylem vessels are actually dead cells, so that they can carry out their functions properly. So these are the different stages where the meristematic cell will undergo the cell division and will result in the formation of these cells that may further undergo dedifferentiation, or they may again enter into the cell cycle by maintaining their meristematic nature. And these differentiated cells acquire a particular function, they become mature, resulting in the formation of mature cell, which will eventually undergo senescence, that is the aging, and the aged cell will ultimately die. So you may write so in the exams, the figure may be given and you may be asked to write the correct process. So you can tell here that the meristematic cells undergo plasmatic growth, and this cell may either enter into the cell cycle or it undergoes differentiation. And meanwhile, the cells undergo expansion or elongation and the mature cells are being formed. These mature cells undergo senescence, resulting in the death of the plant cell. The next is plasticity. So we will learn about what is plasticity in plants. Plasticity in plants is a crucial adaptive mechanism that allows them to respond to thrive in diverse and changing environments. So there are certain plants that initially live in some other environment, but with the which are initially in the water, but then as they become mature, the cells, the plant height becomes long, that it comes out of water. So there are certain mechanisms, adaptive mechanism, for example, that help the plants to respond to and thrive in diverse and changing environments. Through morphological and physiological adjustments, plants optimize their growth and development, ensuring survival and reproductive success across various conditions. So it can be internal plasticity or it can be environmental plasticity. Internal plasticity, in which the leaves of the juvenile, for example, the leaves of the juvenile plant are different in shape from those of the mature plant. So juvenile means young plant. So the leaves of the young plant are different in shape than the mature plant. For example, the heterophyll in cotton, coriander, and larkspur. This is important. The examples are important. Heterophyll is observed in cotton, coriander, and larkspur, in which the juvenile plants are different in shape from those of the mature plants. So this is internal plasticity. On the other hand, differences in shape of leaves produced in air and those produced in water in buttercup. So if I talk about the plant ranunculus, which is buttercup, the leaves which are above the water are different than the leaves which are produced in water. And also present the heterophyllous development due to the environment. So this is environmental plasticity. So these are the figures you can see here, that there is a change in the shape of the leaves of the juvenile and adult plants. The pattern is different. And if I talk about these are the heterophyll in larkspur. The second is the buttercup, where the terrestrial habitat had such big leaves, while the water habitat has different types of leaves. So development heterophyly is also there. In development heterophyly, the leaves of a plant have different shapes and sizes at different development phases of the plant. So as I mentioned, so this is also the internal plasticity. This is also the example of developmental heterophyly, where the shape of the plant is different during the different phases of the development. So when the plant was young, the shape was different. When the plant was adult, the shape was different. So this is known as developmental heterophyly. Second is environmental heterophyly, as I mentioned. This is because of the environment. So environmental heterophyly is found in plants that adapt to the environmental conditions by having different types of leaves in different conditions. So this heterophyly is usually found in aquatics. Example is the buttercup, where the aerial leaves are floating and floating leaves are different. So aerial leaves and floating leaves are different. The growth, differentiation, and development are therefore very closely related events in the life of a plant. Broadly, development. So this is kind of summary of growth and development. That till now, what we have studied about growth and development, that the growth, differentiation, and development are therefore very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. So development is actually happening because of the growth of the cell and the cells undergoing differentiation, acquiring a particular function. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors, that is the internal and the environmental factors. External factors, the former include both intracellular, that is genetic, and intercellular, which is the chemical, such as plant growth regulators, while later includes light, temperature, water, oxygen, nutrition, etc. So these different factors are important in the growth and the development of the plant. So it can be both, the factors can be both extrinsic. So such questions may be asked in MCQ, that which of these factors are responsible for the plant growth and development. So it can be intrinsic or it can be extrinsic. Intrinsic means internal factors. Extrinsic means external factors. Intrinsic factors can be of two types: intracellular, that is within the cell, and intercellular. This intracellular is within the cell and it happens mostly at the genetic level. And intercellular, in order for two cells to communicate, we know that our body usually releases a chemical which is the hormone. So intercellular connections happen. Intercellular factors are the plant growth regulators, which are also known as the hormones that we will see. Right? So growth is the sum of like you can say that the growth, differentiation, and development are very closely related events, and development is the sum of growth and differentiation. So development is a result of both growth, plant growth, and differentiation of the plant cells. So now we will learn about what are plant growth regulators. So plant growth regulators, as I mentioned, they are nothing but they are the hormones that are found within the plants. But they are named as plant growth regulators. So a hormone, what is actually a hormone? So in the original meaning of the term is a signaling molecule that is produced in tiny amounts. Usually it's a signaling molecule. You can define hormone as a signaling molecule that is usually produced in lower concentration by one part of an organism's body and transported to other parts. So one cell produces hormone that act on the another cell. So this is a cell X, this is cell Y. Hormone actually helps in maintaining the communication. So by one part of an organism's body and transported to other parts where it binds to a specific receptor and triggers response in target cells and tissues. So there is a binding of the hormone with the receptor. So on that Y cell, the receptor for the hormone X must be present, that to which the ligand X binds to its receptor. That initiates the cascade of events within the cell. So you can, if I draw it like this, wait. What we can say is, suppose this is the cell, it has the receptor for the hormone X. The receptor will induce a signaling, it will induce signals. The nucleus will get that signal, will start forming the proteins according to that. So in animals, hormones are usually transported. We know that in humans or in animals, the hormones are usually transported through the circulatory system, which is often included in definitions of the term. But many plant biologists, however, argue that hormone concept, which originated from studies of animals, is too limiting to describe plant physiological process. So, according to many plant biologists, the term hormone, which is used in context of the plant, is different than that of animals. For example, we know that in animals, the hormone transport through the circulatory system, but in plants, the circulatory system, circulating blood is absent. For example, plants do not have circulating blood to transport hormone-like signaling molecules. Moreover, some signaling molecules are considered plant hormones, act only locally. So in the definition, we see that these are the signaling molecules that are produced by one cell type and act on the other cells, which are present in another parts of the body, and they are carried out usually by the circulatory system. But in plants, certain examples have been seen where the plant hormones act only locally and they are not carried anywhere. Finally, some signaling molecules in plants such as sucrose typically occur at concentrations hundreds of thousand times greater than that of a typical hormone. So this is also a signaling molecule. Sucrose is also a signaling molecule, but as we mentioned in the definition, that hormone is a signaling molecule that is usually produced in a lower concentration, in a very tiny concentration. But there are certain signaling molecules in plants, for example, sucrose, which is typically occur at a concentration hundreds of thousand times greater. So nevertheless, they are transported through plants and active signal transduction pathways that greatly alter the functioning of plants like a hormone. Thus, many plant biologists prefer the broader term, which is the plant growth regulator. So, plant growth regulators are nothing but they are actually hormones only. But since because of the various limitations or various particular terms which are being used in the definition of hormones, like they are signaling molecules that are produced in very low quantity, as well as they act on the other side, any other body parts, and are carried by they are transported. So, thus, many plant biologists refer the broader term plant growth regulators instead of hormones to describe the organic compounds, whether natural or synthetic, that modify or control one or more specific physiological process within the plant. So there are examples like indole compounds, which is indole 3 acetic acid IAA, adenine derivative, which is N6 perfluoryl aminopurine, which is kinetin, derivatives of carotenoids, that is abscisic acid ABA, terpenes, gibberellic acid GA3, or gases ethylene. So we will mainly learn about these hormones, plant hormones in literature, which is IAA, indole acetic acid, or auxin, if I say. We will learn about gibberellic acid GA. We will learn about ethylene. We will also learn about abscisic acid ABA, and we will learn about kinetin, which is a type of cytokine. So these are the different plant growth regulators which are present in the plants. And they are usually the indole compounds, for example, the indole 3 acetic acid, or they are the adenine derivatives, that is N-perfluoryl aminopurine kinetin, derivatives of carotenoids, abscisic acid, etc., terpenes, gibberellic acid, etc., or gases. Now, what are the characteristics of plant growth regulators? So plant hormones are produced in very low concentrations, but a tiny amount of hormone can profoundly affect plant growth and development. We know that the hormones are usually present in tiny amount, but that tiny amount is enough and has a great effect on the growth and development. Virtually every aspect of plant growth and development is under the hormonal control to some degree, and each hormone has multiple effects depending on its site of action, concentration, and the plant's developmental stages. Conversely, multiple hormones can influence a single process. So, both of these things can happen. A single hormone may carry out many functions, or different functions are regulated by the single hormone. Both of these things may happen. It may carry out function 1, function 2, or function 3, etc. Response to hormone usually depends not so much on the amount of that hormone as on its relative concentration compared with the other hormones. So, individual amount does not affect that much as compared to the relative concentration compared with other hormones. It is often the interactions between the, why it is so, because it is usually the interaction between different hormones rather than the hormones acting in isolation that can control the growth and development. Therefore, the interaction between the hormones is necessary, as the growth or the responses of the hormone usually does not depend on the concentration of a single hormone, but on the relative concentration of the compared with the other hormones. Why it is so? Because it is often the interactions between the different hormones rather than the hormone itself acting in isolation that control the growth and development. So these are the characteristics. The PGRs can be broadly, the plant growth regulators can be broadly divided into two groups based on their functions in a living body. So based on the function, PGRs can be divided into two groups. First is which are involved in promoting activities such as cell division. So basically the first one are the ones, if I would like to write. So first one are the ones that are playing an important role in growth. And the other one are the one that helps during the stress condition. So the one group of the PGRs are involved in the growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are called the plant growth promoters. Example: auxins, gibberellins, and cytokinins. Example: auxins, indole compounds, indole 3 acetic acid, auxin, adenine derivatives, as I mentioned, N6 perfluoryl aminopurine, which is the kinetin, which is a cytokinin. So this is a cytokinin. This indole 3 acetic acid is auxin, and terpenes, which are gibberellic acid, GA3 is gibberellin. The PGRs of the other group play an important role in plant responses to wound and stress. And they are also involved in various growth inhibiting activities. So first one is the growth promoting activities, plant growth promoters. They are plant growth promoters, plant growth inhibitors. Plant growth inhibitor. This is important. These are the two classes. One that that helps in the growth. The second one is that helps in the inhibition, such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but is largely an inhibitor of growth activities. The example include gaseous ethylene and the derivative of carotenoid, which is abscisic acid ABA. So one plant growth hormone that is gaseous in nature. What is the example of this? The example is ethylene. This might be asked. One plant growth hormone which is gaseous in nature, that is ethylene. T2H4. So these are the different structures of different. You can see that it has indole ring, auxin. Then we have cytokinin. Example include kinetin. We have polyamines, we have abscisic acid, we have nitric oxide, salicylic acid, jasmonic acid, brasinosteroid, ethylene, which is a gaseous. So ethylene is a gaseous PGR. And we have gibberellins. So these are the different plant growth regulators. We will mainly focus on the ones which are mentioned in NCERT: gibberellins, auxins, cytokinins. We will focus on abscisic acid and we will focus on ethylene. And in the last, we will focus a little bit on the brasinosteroid as well, because questions are asked from this portion also. So before proceeding further, we first need to understand the discovery of these plant hormones. The discovery itself the discovery of these hormones are accidental, and the discovery itself gives an idea about the function of these plant hormones. So the idea that the chemical messengers exist in the plants emerge from a series of classic experiments on how stems respond to light. The shoots of the plant grows toward light. This is very important. So suppose if this is any plant and the direction of light is this, that the shoot of the plant will grow towards this. What's the direction of the light? So the Greek word, this is any growth response that result in plant organs curving toward or away from the stimuli is called the tropism. Tropos means turn. So it is example of phototropism. Photo means light and bending towards the light. So photo means light, tropism means bending towards. So the growth of the plant organ towards light or away from it is called the phototropism. That is the growth of a plant organ that is towards the light or away from it is called the phototropism. Shoots generally exhibit positive phototropism. So that is they grow towards the light, whereas roots exhibit negative phototropism, grow away from the light. While the shoots grow towards the light. In nature, positive phototropism directs shoot growth towards the sunlight that powers the photosynthesis. This response involves cells on the darker side. So why this is happening? How this must be happening? When I say that the plant, suppose this is the plant shoot. And when I say the direction of light is this, and ultimately it is bending towards this. So this is the light portion, this is the dark portion, right? That is the portion away from the light. So how does this bending might happen? So the bending might happen because of the elongation of the cells, on darker side. So when these cells elongate, cells which are not receiving any light elongate, they curve and bend. So this response involves cells on the darker side elongating faster than the cells on the brighter side. So Charles Darwin and his son Francis conducted some of the earliest experiment on phototropism. So we will see what was the experiment that was being conducted. So in this experiment, they carry out 4 sub-experiments in which In the first one, there was a shoot which is exposed towards the light, and we can observe what we can observe is the bending towards the light. You can see that the shoot is bending towards the light. But in the another experiment, when they cut the shoot apex is cut. You can see here that they cut the shoot apex. So they cut the shoot apex and they found no effect of light. There is no bending, so no effect of light. That is, phototropism is not observed. In the third case, they cover the shoot tip with the opaque cap. So this opaque cap prevents interaction with the light, and therefore bending does not happen. Bending does not happen. And in the fourth, a transparent cap is being put. Transparent. So the transparent cap will allow the light to enter and interact with the cells. Therefore, what we will see is the bending. So the question may ask that you have to clearly observe that in which of the following, in any of the experiments, in such phototropic experiments that was performed by Charles Darwin and his son Francis, you must see that the tip of the shoot should get a chance to interact with light. Only then the bending will happen. So if we have to ask, in A, bending will happen? Yes. In B, it will not happen. In C also, it will not happen. So in B and C, the bending will not happen. But in D, bending will happen. So in A and D, where the tip. So what conclusion can we draw from this? That definitely there is a role of this coleoptile, that is the tip region of the shoot. So it was the tip of the coleoptile, the Darwins concluded, that was responsible for sensing the light. Because once we cover it with the dark structure or once we cut it, the interaction with the light hampered and we did not notice any bending. However, they noted that the differential growth response that lead to curvature of the coleoptile occurred some distance below the tip. So you can see here that if I cover this with the transparent ones, you can see that there is at a certain distance below the tip, there is a curvature that led to the curvature of the coleoptile occurred some distance below the tip. So Darwin's postulated that some signal was transmitted downward from the tip to the elongating region of the coleoptile, and auxin isolated. So this signal was actually the auxin, which was the signal was actually through the hormone or the plant growth regulator through the auxin, and this was isolated by FW Wendt from the tips of the coleoptiles of the oat seedling. So this is important. The auxin was isolated by FW Wendt from the oat seedling. The next, the discovery of cytokinase. So cytokinase, Cook and his co-workers observed that from the internodal segments of tobacco stem cells, the callus, which is a mass of undifferentiated cells, we know that callus is formed as a result of dedifferentiation, that is, it carries undifferentiated cells. Dedifferentiation. That is, it carries undifferentiated cells and proliferate. These cells proliferate only if, in addition to auxins, the nutrients medium was supplemented with one of the following extracts. That is, if I provide auxins with nutrient mediums to the extract of vascular tissue or yeast extract, coconut milk, or DNA. So Skoog and Miller 1955 later identified and crystallized the cytokinesis promoting active substance. So since this substance was promoting cytokinesis means division of cells. And since this was promoting cytokinesis, therefore they were named as cytokinins. Therefore, they were named as cytokinins, and it was discovered by Skoog and Muller in 1955. It was identified by Skoog and Muller in 1955. The third one is the gibberellic acid. So, first cytokinin that was discovered that they identified was termed as the kinetin. Then third one is the gibberellic acid. So, in early 1900, it was observed that the Asian farmer noticed that some rice seedlings in their paddies grew so tall and spindly that they toppled over before they could be mature. So before maturation, they become tall and toppled over before they become mature. So in 1926, E. Kurosawa discovered that this is due to a fungal infection due to the fungus of the genus Gibberella fusicorae. Genus Gibberella, species fusicorae, causes this foolish seedling disease. So the disease is known as the foolish seedling disease. So by the 1930s, it was determined that the fungus caused hyperelongation of rice stem by secreting a chemical called gibberellin. So in 1926, E. Kurosawa discovered that this is caused by a fungus which is Gibberella fusicorae, and in 1930 it was determined that the fungus is responsible for this hyperelongation because it is secreting a chemical which is the gibberellin. In the 1950s, researchers determined that plants also produce gibberellin, which is a gibberellic acids. These are known as gibberellic acids, and many gibberellic acids have now been identified. So this is how the accidental identification of the gibberellic acid happened. The last that we will discuss is the abscisic acid. So during the mid-1960s, three independent researchers reported that the purification and chemical characterization of three different kinds of inhibitors, that is inhibitor B, abscission 2 and dormant. Later, all three were proved to be chemically identical and it was named as abscisic acid. So these were the inhibitors. So these 5 plant growth regulators are very important. The first one is auxin, which is plays an important role in the stem elongation. The second one is the cytokinins that are playing an important role in cytokinesis. The third one is gibberellic acid that is playing an important role in the stem elongation. And the fourth one is abscisic acid, and the last one ethylene. So H. Cousins in 1910 confirmed the release of a volatile substance from ripened orange that hastened the ripening of the stored unripened bananas. So this hastened the ripening of the stored unripened bananas. So what was responsible for this? Ethylene was responsible for this, which is gaseous in nature and is volatile. Gaseous and is volatile. So he confirmed the release of this volatile substance from the orange that were already ripe. Ripened oranges were releasing some volatile substance that hastened the ripening of stored unripened bananas. So you might have seen even at homes also, if there is some unripe fruit and if we place it near the ripened oranges or ripened apples, then these fruits become the unripened fruits rapidly ripe. So why this is happening? Because of the chemical signal, which is the ethylene, so which is gaseous in nature and is volatile. So later, this volatile substance was identified as ethylene, a gaseous plant growth regulator. So these were the 5 different types of plant growth regulators that are majorly in focus, and we will discuss majorly on these 5 plant growth regulators and their functions. So what are their functions, how they are helping in that function, and what is their role in the plant development. So thank you for attending today's session. We'll discuss about these PGRs in detail in the next session. Thank you.